طلبتنا الأعزاء، السلام عليكم. محاضرتنا لهذا اليوم عن البكتيريا العائدة إلى جنس الباسيلس. The genus Bacillus include large gram-positive rods occurring in chain. Most members of this genus are saprophytic organisms prevalent in soil, water, and air, and on vegetation, such as Bacillus cereus and Bacillus subtilis. Bacillus cereus considered as an opportunistic pathogen. Bacillus anthracis, which causes anthrax, is the principal pathogen of the genus. The typical cells are a large gram-positive rods, have square ends, and are arranged in long chains. Spores are located in the center of the non-motile bacilli and has the same width of the bacilli. This bacteria, when cultured on blood agar colony of Bacillus anthracis, are round and have a cut glass appearance in transmitted light. Hemolysis is uncommon with Bacillus anthracis, but common with the saprophytic bacilli such as Bacillus cereus. This feature helps in the differentiation between two species. Gelatin is liquefied. In human, the infection is usually acquired by the entry of spores through the injured skin in case of cutaneous anthrax, or rarely the mucous membranes of gastrointestinal tract cause gastrointestinal anthrax, or by inhalation of spores into the lung in inhalation anthrax. The spore germinates in the tissue at the site of entry, and the growth of the vegetative organism then produced toxin result in the formation of gelatinous edema and congestion. Bacilli spread through the lymphatics to the bloodstream, and they multiply freely in the blood and tissue shortly before and after death. This is very important note. Bacillus anthracis that does not produce a capsule is not virulent, so the capsule play an important role in the pathogenesis of this bacteria because of this capsule is antiphagocytic. Capsule of Bacillus anthracis is distinct between bacterial capsules because of it's composed from poly-D-glutamic acid. This means has a protein in nature, while the bacterial capsule of other genus is composed from polysaccharide. Anthrax toxin also distinct and unique between bacteria because of it composed from 3 subunits, while the other bacterial capsule is composed from 2 subunits, binding and active subunit. In case of anthrax toxin, is composed from protective antigen, which act as a binding subunit, edema factor, and lethal factor. Protective antigen binds to specific cell receptors, as seen in the figure below, and following proteolytic activation, it formed a membrane channel that mediated the entry of edema factor and lethal factor into the cell. Edema factor is an adenylate cyclase make to formation of cyclic AMP from ATP through the calmodulin channel. This leads to accumulation of fluid and electrolyte inside the cells, while lethal factor plus protective antigen form the lethal toxin, which is help in the destruction of the cells and lead to cell death through the activation of MAPIC pathway, mitogen activation protein kinase. In a human, approximately 95% of cases are cutaneous anthrax and 5% are inhalation. Gastrointestinal anthrax is very rare. Cutaneous anthrax generally occurs on exposed surfaces of the arms or hands. A pruritic papule developed after 1 to 7 days of entry, the papule rapidly change into a vesicle that coalesces and a necrotic ulcer develops. Lesions have a characteristic central black eschar. Marked edema occurs, lymphangitis and lymphadenopathy, and systemic signs including fever, malaise, and headache may be occur. After 7 to 10 days, the eschar is fully developed. It dries, loosens, and separates. Healing is by granulation and leaves eschar. In as many as 20% of patients, cutaneous anthrax may be developed to the sepsis, systemic infections, including meningitis, which lead to death. The incubation period in inhalation anthrax may be as long as 6 weeks. Hemorrhagic necrosis and edema of the mediastinum substernal pain may be prominent. Hemorrhagic pleural effusion, cough, sepsis occurs, and there may be hematogenous spread to the gastrointestinal tract, which lead to bowel ulceration, or to the meninges, causing hemorrhagic meningitis. The fatality rate in inhalation anthrax is very high. For diagnosis of this bacteria specimen, including fluid, pus from lesion, blood, and sputum, this stained with gram stain or immunofluorescent staining technique, which can be used to detect anthrax bacilli. This bacteria cultured on blood agar plate. Anthrax bacilli produced non-hemolytic gray to white colonies with a rough texture and a ground glass appearance. Coma-shaped outgrowth, like medusa head, may be projected from the colony. In semi-solid medium, anthrax bacilli are non-motile, whereas related non-pathogenic organisms such as Bacillus cereus exhibit motility by swarming. This distinct feature helps in the differentiation between two species. Now, to differentiate between Bacillus anthrax and Bacillus anthracis can be used motility feature and the appearance of colony and hemolysis. Bacillus anthracis is not hemolytic, while Bacillus cereus hemolytic and motile. Also can be used ELISA technique for determination of antibody against edema and lethal toxin, and can be detected of this bacteria by using PCR, but this test is very rare and can be used in reference laboratory. Now, treatment of infection. Many antibiotics are effective against anthrax, like ciprofloxacin is recommended for treatment of anthrax. Penicillin G in combination with gentamicin or streptomycin as used as alternative drugs. In the setting of potential exposure to Bacillus anthracis as an agent of biological warfare, prophylaxis with ciprofloxacin or doxycycline should be continued to 4 weeks. Some other gram-positive bacilli, such as Bacillus cereus, are resistant to penicillin by production of beta-lactamase, so doxycycline, erythromycin, or ciprofloxacin may be effective alternatives to penicillin. Epidemiology, prevention, and control of anthrax infection, including the soil is contaminated with anthrax spore from the carcasses of dead animals. These spores remain viable for decade. So the control measure, including disposal of animal carcasses by burning or depural and lime pits, decontamination of animal products, protective clothing and gloves for handling potential infected materials, and active immunization of domestic animals with live attenuated vaccine. Now, Bacillus cereus is considered as an opportunistic pathogen. This bacteria causes food poisoning, which present in two forms: the emetic type, associated with fried rice, and the diarrheal type, associated with meat dishes. Bacillus cereus produces toxin that cause disease that is more an intoxication than a foodborne infection. Emetic form is manifested by nausea, vomiting, abdominal cramps, and occasionally diarrhea, and is self-limiting, with recovery occurring within 24 hours. It begins after 1 to 5 hours after ingestion of rice and occasionally pasta dishes. Bacillus cereus is a soil organism that commonly contaminated rice. When a large amount of rice are cooked and allowed to cool slowly, the spore is germinate and the vegetative cells produce the toxin during log phase of growth or during sporulation, while the diarrheal form has an incubation period of 1 to 24 hours and is manifested by profuse diarrhea with abdominal pain and cramps. Fever and vomiting are uncommon. The enterotoxin may be produced in the food or in the intestine, but the presence of Bacillus cereus in the patient's stool is not sufficient to make the diagnosis of Bacillus cereus disease. Also, Bacillus cereus is an important cause of eye infections, such as severe keratitis. Typically, the organisms are introduced into the eye by foreign bodies associated with trauma. Also, Bacillus cereus associated with localized infection and with systemic infection, including endocarditis, meningitis, osteomyelitis, and pneumonia as an opportunistic pathogen. Thank you for